Chapter 13. This is viruses, viroids, and prions. So, since we're dealing with some very, very, very small things, if you cannot do great things, do small things in a great way. That's kind of how we can start out. The father of virology, Martinus Bajaric. That's as close as I'm probably going to get to such a unusual spelling name. Alright, so we're going to get started with chapter 13, where we're going to be talking about a little bit of history and things like that. Really get into what a virus is. Now, viruses, the thing about viruses is when we say they're not alive, they break the rule of life, the things that living things do, but they evolve from living things, it has appeared. They are obligatory, means they have to be intracellular parasites. They require a living host to actually multiply. They cannot do the basic life things. They don't have any enzymes for metabolism anyway. They do have enzymes, it's not enzymes for metabolism. They don't do protein synthesis on their own. They can't make ATP. So they are basically not alive until they're in you. But they're really weird. They appear to have evolved from living things. They contain DNA or RNA. They are single or double-stranded DNA or RNA-based diseases. It just depends upon which virus you're dealing with. They have a protein coat called a capsid. Some have a lipid envelope or a protein envelope or a carbohydrate envelope. Envelope viruses are really good, like lipid envelope viruses, like SARS-CoV-2. Washing your hands for the envelope virus can make it a little bit easier to have it. They have spikes, which are attachment identification. The spike proteins are what the SARS-CoV-2 vaccine, the COVID shot, especially the Pfizer mRNA-based vaccines, were spike proteins. They coded it was mRNA that can be translated into a spike protein. Okay. Most viruses can only affect specific types of cells in one host. So what I mean, like, it might just infect a kidney cell. Sometimes they can be a little bit more generalized. It depends upon what it is. What kind of host cells can it, even what kind of host. It might be very specific to cold-blooded hosts. It might be very specific to warm-blooded hosts. Mammals only, things like that. So sometimes it affects only dogs. So there is a host range, a spectrum of host cells the virus can affect, determined by the attachment sites and cellular factors that cell has. Viruses can make structures to transport viral nucleic acids to other cells, but they can be very specific. Viruses are quite small. Here we have a normal red blood cell. Here we have an E. coli, and then we have something like Ebola. These things are some of these viruses, like the elementary body of chlamydia, which is the secreted from it, pretty small. Some of these are smaller than light, like 90 nanometers. That's much smaller than light. There are like some really famous viral shapes, like the rabies virus looking like a bullet, Ebola virus, the shepherd's crook, the bacteriophage, a T4 bacteriophage, very famous. Bacteriophages are the deadliest virus known, kills more. There's all kinds of things there. Anyway, virions, this is a complete and fully developed infectious viral particle and has some nucleic acids. The nucleic acid could be a DNA or an RNA. The capsid is the protein coat. It's for protection. It's like a vehicle for transmission that we can sneeze and the virion goes out. If it's enveloped, some of them are enveloped viruses. Some of them are non-enveloped viruses. There may be spikes. Viruses are classified by differences in the structures of their coats. For example, here we have an envelope virus who has an envelope with spikes, indicative of things like influenza, so washing your hands is really good. helical viruses, for example, like Ebola, the famous shepherd's crook Ebola shape. The T-even bacteriophages, things like that. This virus down there looks like a nice little smallpox. Looks like a lovely little smallpox, a very scary little virus. Viruses do have taxonomies because they evolve from living things. Their family name ends in something Viridae. The genus name is something virus, like adenovirus, influenza virus. Okay, viral species sharing the same genetic information and have the same ecological niche, which in this case, the niche for a virus is the host. Common names are used. For example, we might have herpes, but we have herpes viridae, herpes virus, humans herpes virus, and then we have other like HHV1, HHV2, and HHV3. HIV is retroviridae, lentivirus, human immunodeficiency virus, HIV, HIV1, and HIV2. Okay, the bacteriophage is really the deadliest virus on planet Earth, is how it has been told. It is, they kill a lot of bacteria, so they wipe bacteria. We can wipe out a lot because there's a lot of them. They attach to the fimbriae or even the flagella, so flagella is sometimes a target for these viruses. One of the things we use is bacteriophage to treat these, called phage therapy. Now, viruses have two different parts of a life cycle. Two different kinds of life cycles for viruses, lytic and lysogenic. Now, this is important for us because different viruses that go dormant and can then come back in a different way. For example, things that like dormant, like herpes viruses. This has to do with their life cycle, and also the zoster virus, things like that. Lytic life cycle causes death of the cell. The cell comes in, it injects its DNA, the cell bursts, and then we spread virus everywhere. Lysogenic cycle is the host cell stays alive, and actually the phage will incorporate its DNA, and the host cell has new properties. Phage is actually what we call a phage. Phage DNA incorporates in the host cell, phage conversion, and then a specialized transduction DNA, the new cell, we call that a prophage. Okay, so basically we can attach it, it shoots its DNA, the DNA comes in and hijacks the cell's molecular machinery, and the cell starts making virus bits, and then what happens is the virus pops, dies, killing the host cell. So that's what we call lytic life cycle. Lysogenic, it incorporates itself into the host. It makes itself part of the DNA, and so that's the difference between lytic and lysogenic. Lysogenic doesn't kill you. Now, animal viruses, they have to first attach. Okay, now that's virus attaches to the cell membrane. Now, we have to penetrate, we have to uncoat, then we go through biosynthesis, maturation, and release. Attachment viruses attach to the cell membrane. We penetrate by endocytosis or fusion. So sometimes, for example, in animal viruses like the herpes virus, the pressure is so great inside of a herpes virus, it is higher than the pressure in your car tires, and it just shoots that DNA into you. We can uncoat by viral host enzymes. Then we start producing nucleic acids and proteins of the virus. The virus matures, nucleic acid and capsule proteins are assembled, and then we can release or rupture new viruses. Depends on if it's the lytic or lysogenic cycle, the virus is released. So basically the virus comes in, it attaches, it shoots its DNA in, the DNA comes in and hijacks your cell's molecular machinery. The cell starts becoming a virus factory, and then it starts making new viruses that can spread, and the virus spreads and spreads and spreads. Okay? So it can bud and release using budding to make an envelope virus. Okay? Now, viral infections can be asymptomatic for a long time, like with cold sores and shingles, where there's a latent viral infection. The latent infection is what happens after years. You got the virus that causes chickenpox, and then years later you go through a period where you have a depressed immunity, like you get really tired, you get sick. It knocks your immune system down a peg, and you start getting the virus taking over. The measles virus, for example, disease can processes occurs over a long period, and it can be very fatal. Subacute sclerosing panencephalitis, a measles virus, can do this. Okay, where it can be persistent and then get you. Okay. Now, one of the things that viruses can do that's really terrifying about viruses is viruses can come in and rewrite your DNA. We either have Ebola DNA in our genome, or Ebola has human DNA in its genome. We can do this. Oncogenes, these are genes that bring about transformation into cancerous cells. They are genes that transform a normal cell into cancer cells. An oncovirus, or an oncogenic virus, is a virus that can cause tumors in animals. The genetic material becomes integrated in the host cell's DNA and changes the expression of a gene. Sometimes these genes are needed to control the cell cycle, like mitosis. And it might be, for example, a gene that gets mutated, that gets altered, where this DNA is integrated into the host DNA, and it changes the gene, and it makes the cell cycle not work right, and the cells grow uncontrollably and don't stop growing, and we end up with what is referred to as a cancer. Okay, and 10% of cancers are viral. A good example of this is human papillomavirus. We used to think, because of this, that viruses were the cause of all cancers, but there are mutations, carcinogens, chemical exposure, DNA, the DNA damaging chemicals that cause mutations. Okay, for example, stuff in cigarette smoke, benzene, things like that. We can culture viruses. They must be grown in living cells. They need a special chemical medium. Medium where there's a bacteriophage and a bacteria who are together, for example. Several viral replication cycles might happen. The bacteria that surrounds the original virus gets destroyed, and that makes plaques. And the plaques is clearing a bacteria along because the bacteriophage is destroyed, lysed the cell. So hopefully we have each plaque is one virus. Just like we have colony-forming units, we have plaque-forming units. We can also grow viruses in a laboratory. We can use living animals to study immune responses. For example, we can shoot the virus into a rat. We have embryonic eggs, like chicken eggs, that are used to grow vaccines. This is why we will have certain egg sensitivities, egg allergies. There we go. Can't take certain vaccinations. There's cell culture, which is the preferred medium. It's just convenient. It's a cell culture medium. Fetal bovine serum and some of that, and that's what I did. It's the only way I ever really grew viruses, was in cell culture media. I never got to do living animals or embryonic eggs. Prions, which stands for proteinaceous infectious particle. Prion is actually a disease caused by misfolded proteins. It's a protein that is causing disease. Okay, it's inherited by ingestion, transplant, or by surgical instrument. There are spongiform encephalopathies called sheep scrappy, Creutzfeldt-Jacobs disease, and then one that was newer to me was Gertzmann-Straussler-Schenker syndrome. I believe that's how you say Schenker. I think it's Schenker syndrome. Fatal familiar insomnia, mad cow disease. Creutzfeldt-Jacobs, and there's a variant where it's transported by bites, basically zombies. Mad cow disease became mad human disease, became mad zombie disease. A normal cellular prion protein on the cell surface is PRPC. PRPSC is a scrappy protein in brains, forms plaques we call sheep scrappy. These cause encephalitis, brain holes eaten in the brain, bovine spongiform encephalopathy. So, for example, these cells produce that misfolded protein, an altered gene, a mutation. It reacts with stuff on the cell surface. It gets converted. It's then taken in, and then they accumulate in the cell till the cell dies, because it creates these kind of accumulations of proteins inside and becomes disease-causing. So that's how it causes disease, basically. Okay. Plant viruses, a good example of a plant virus is tobacco mosaic virus, potato spindle tuber disease caused by a plant pathogen. Viroids are infectious RNA. They're plant pathogens. There's no protein code. It doesn't code for proteins, but it causes a disease. They can enter through insects or a wound in a plant. Tobacco mosaic virus is a good example of that. All right. There we are, and we'll see you in the next one.